every cubic centimeter of urine formed, about 1,000 milliliters of blood pass through the kidneys. The kidneys are the principal functional organs of the urinary system. One kidney can be lost, donated, surgically removed, without seriously impacting renal function. In fact, about 0.04% of the population is born unirenal. Since they are hung on the posterior body wall and extend below the lower ribs, the kidneys are vulnerable to contusions from sharp blows to the lower back. This is the anatomical basis for the kidney punch of boxing. The fibrous attachment to the posterior body wall dictates that a kidney has peritoneum only on its anterior surface. For this reason, the kidneys are said to occupy a retroperitoneal position, literally behind the peritoneum. This is a distinction that they share with only a few other organs, adrenal glands, and the abdominal parts of the aorta and inferior vena cava. By forming urine, the kidneys play important roles in a number of homeostatic mechanisms.
Hi, I'm Mike Shively. Welcome to Introduction to Anatomy and Physiology. We're talking about the urinary system. Anybody have any questions from last time on the urinary system, Susan?
Well, I had a question on the intro. They had talked about the kidneys being retroperitoneal, and what does that mean exactly?
Yeah, let's go back to, we talked about this very briefly, but I probably didn't explain it good enough. Let's go to the overhead camera for just a second. And I don't know if you recall, but when we were talking about the digestive system, we drew a kind of an egg-shaped thing. We said this is a transverse section through an abdomen. And we probably put even a kidney about here and a kidney about here and some other organs like intestine down in here. And then we said what the lining serous membrane does in the abdominal cavity is it coats the inside of the abdominal wall like I'm doing with this red line. Comes up and goes around the kidneys like this. Then it'll come down and go around a loop of intestine and go back and come down and go around another loop of intestine and go back and so forth. And we said this layer out here, just on the body wall, was parietal peritoneum. Do you remember that?
Yes.
Us saying that? And then the peritoneum that's actually on the surface of an organ, like this intestine or that kidney, is called visceral peritoneum. And this was the peritoneal cavity. I'm just going to write P cavity here. Now, most organs in the abdominal cavity are actually out in the middle of the space someplace, and as a consequence, they have peritoneum going around the whole thing. Like this piece of intestine has peritoneum really on all sides of it. But a few organs, like the kidneys, and I think it was also mentioned on that pre-taped video segment, the adrenal glands, the abdominal parts of the aorta and inferior vena cava, those organs are actually plastered against the posterior abdominal wall, and as a consequence they only have peritoneum on their front sides. Peritoneum does not go around on their back sides. And organs that have that kind of positional relationship with the peritoneal cavity, where they only have peritoneum on the front side because the organs themselves are kind of glued, if you will, by fascia to the posterior body wall, are said to be retroperitoneal. That basically means behind the peritoneal cavity. Retro, behind, yeah. Starley?
I know this goes back a session or two, but I was wondering if you could go over the biliary duct system again.
You know, I may not have even gone over it, Starley. Let's stay on this overhead camera for just a second, and let's let this be the liver. We'll just draw a big old arc, half-moon thing. We'll say this is the liver. Now, coming out of the liver, the two main biliary channels that leave the liver are the left and right hepatic ducts. So we'll let these be the left and right hepatic ducts, and they actually come down here and join to form a single common hepatic duct at this point. So that's a hepatic duct. And this is another hepatic duct over here, left and right. And where these come together, this is called the common hepatic duct. Now, coming on down just a little ways, we've got another channel coming in to the common hepatic duct from the side, and that channel actually goes up and has the gallbladder on it. This duct is called the cystic duct. Now, when the common hepatic duct and the cystic duct join, the biliary passage from that point on down is called the bile duct. In a lot of texts, you'll see this called the common bile duct, but there's no reason to call it common bile duct. The official name is simply bile duct. Now, the bile duct goes on down. Coming into it, we actually have, I'm going to use a different color here. We actually have the pancreatic duct joining into it. So I'm just going to write a green arrow there. I don't know how well that's showing up. I'm going to write from pancreas. How's that? From the pancreas. So this is the pancreatic duct. Now where the two join, that common channel, and I'll use a different color from this point on so that we indicate that we've changed the name. That common channel is actually called the hepatopancreatic ampulla right here. And that actually goes into the small intestine and opens within the lumen of the small intestine on a little tiny bump, a little papilla, if you will, that's called the major duodenal papilla. So this is in the descending part of the duodenum. So bile, when it's formed by the liver, of course, comes out of these hepatic ducts. And if there's a little smooth muscle sphincter down here, if that smooth muscle sphincter is clamped down, that bile, as the pressure builds up, will simply go up the cystic duct and be stored temporarily here in the gallbladder. Then when bile is needed and this little sphincter muscle down here relaxes, that bile can then be expressed from the lumen of the gallbladder right on down the bile duct and come on down into the lumen of the small intestine. And when it gets in there, what it does is help emulsify lipids in that digestive process. And again, this same common final channel, it's not important to me that you remember that, hepatopancreatic ampulla it's called, actually carries in pancreatic juice from the pancreatic duct. Now the accessory pancreatic duct, if you'll remember there were two pancreatic ducts, and that accessory pancreatic duct makes its own way into the lumen of the descending part of the duodenum and opens at another little tiny projection called the minor duodenal papilla. At any rate, let's go back to the overhead camera just one more time because I want to show what happens if an individual has their gallbladder removed. It's typically because biliary precipitants called gallstones or bladder stones are precipitating in the bile, and as long as they remain up here in the gallbladder, they don't cause serious problems. But when they migrate down through the cystic duct or get into the main bile duct here, they can cause severe pain due to the stretching action and can even cause blockage of the whole duct system. And so for that reason, you know, when people have difficulty, quite often the gallbladder will be removed. Surgeon will go in here and actually tie this off right here and remove the gallbladder. The individual can still make bile, the liver is still making bile, and that bile can still get into the lumen of the small intestine. The bile simply is not stored in the gallbladder. Some individuals that have had their gallbladder removed report that they have more difficulty eating fatty materials, and you know, like fried chicken and pizza are no longer on the agenda because of indigestion problems. Other individuals that have had their gallbladder removed report that they don't have to change their diet didn't have to change their diet significantly at all, and they can eat about the same things that they did before. Did that clarify anything on the biliary duct system that you may have had? I don't believe we covered that, so I'm glad you asked. Any other questions on anything? Amber?
Would you mind explaining the difference between a nephrectomy and a nephrotomy?
Let's put those words up on the screen for everybody to see. Let's go to the overhead camera. You asked nephrectomy versus nephrotomy, right? Nephrectomy is removal of a kidney. So if we're going to take a kidney out, that's called nephrectomy. The ectomy part of the word, the suffix, implies removal. So we can do that same kind of thing to a lot of different organs, of course. If we have a nephrotomy, we're talking about an incision into a kidney. Like one might do a nephrotomy, for example, if you have a very large renal calculus, where we've got precipitation in the renal pelvis and right at the beginning of the ureter. A surgeon might do a nephrotomy to open that kidney up so that that calculus material can be physically removed. Let's go ahead and take a look at a couple of other words. Let's go back to the overhead camera. If we have something that's an osis, that implies a condition of, but it's not specific. Like a nephrosis is a renal condition, but we're not saying what the particular disease is. And I think probably all of you know that if we have something that's an itis, the implication here is that we've got inflammation of that particular structure. So a nephritis would be an inflammation of a kidney. Does that help on those?
Yeah, thank you.
Sure. Well, we want to talk about renal physiology a bit today, and we talked about the gross aspects of an anatomy of the urinary system last time. We need to get down to the functional subunits of the kidney that are actually producing urine. And those subunits are called nephrons. Let me write that word up on the overhead. These are very, very small. There's about 1 million of them in each kidney, roughly 1 million. Now, nobody's ever sat down and counted those one at a time, of course, but about 1 million in each kidney. So they're quite small, and it actually are 3 parts to each nephron. Let's go to a graphic that will summarize the components of a nephron. The 3 components, the 3 main components, the large subdivisions, are called glomerulus, glomerular capsule, and renal tubule. Glomerulus is actually a tuft, if you will, of blood vessels. They're kind of halfway in between arterioles and capillaries. Probably best to think of those glomerular vessels as arterioles. The glomerular capsule is a membranous encapsulation around that tuft of blood vessels, and leading out of the glomerular capsule we have a renal tubule that actually has 4 main subdivisions and several smaller ones. What I would like to do to explain this is to have us all get involved in a little pencil and paper exercise to actually draw a nephron. So let's go to the overhead camera, and you do this. I like to draw glomeruli because I'm actually pretty good at it. Want to see it? Watch this. How's that? Good. Nice glomerulus, huh? I'm going to put 2 arrows on this to indicate that's the arteriole bringing blood into this tuft of vessels, and another arrow here saying that's the one that's draining the blood back out. So this is called the afferent arteriole, and this one is called the efferent arteriole. The glomerulus itself is this little tuft of vessels. Now the glomerular capsule actually comes around the glomerulus, and there's an inner layer that's right on the surface of those glomerular vessels, and then there's an outer layer of the capsule. So we call this the internal layer of the glomerular capsule, sometimes called visceral layer, and this is the external layer of the glomerular capsule, sometimes called the parietal layer. And where I'm putting these red X's is actually the lumen, if you will, the space of the glomerular capsule. Now, the renal tubule is very long and has some named subparts. The initial part is kind of tortuous, goes back and forth a bit, turny and twisty. And then there's a part that comes down and makes a long hairpin loop. And that actually has a skinny part and a fatter part and so forth. And we'll just draw a hairpin turn here, a long straight hairpin turn. And then there's another turny twisty part. And then that dumps into another portion of the tubular system over here. Now I'm going to label this number 1. This hairpin loop I'm going to label number 2. This turny twisty part up here we'll label 3. And this part over here we'll label 4. Okay? This first part, coming right off the glomerular capsule, the initial part of that renal tubule, is actually called the proximal convoluted tubule. Let me see if I can get all that in here. Proximal, meaning initial or first part. Convoluted, implying turny and twisty. And then tubule. Proximal convoluted tubule. The second part, that consists of this hairpin loop, is the nephron loop. Now, in a lot of texts, you'll see this labeled by, it's an old eponym, which was loop of Henle. So you'll see it called loop of Henle a lot. Henle. But its preferred name is just nephron loop. Number 3 is the distal convoluted tubule. And the fourth part over here is the collecting duct. All four of these together, all four of these parts together are one renal tubule, if you will. So the renal tubule has these 4 main subdivisions. Now, there's some other subdivisions. I don't want you to learn these, but I want to mention them to you so that you won't get confused by the literature. When you take a look at this nephron loop, there's actually 3 named parts of the nephron loop. There's what's called a proximal straight tubule to it. Then there's a real skinny part called the attenuated tubule. And then there's a distal straight tubule. All 3 of those things are parts of the nephron loop. So I'm going to say that this has 3 parts to it. But you need not learn those. But when you hear the name like proximal straight tubule or attenuated tubule or distal straight tubule, know that they're talking about parts of the nephron loop. Same thing when we get down here to the collecting duct. There's actually some named parts to the collecting duct. The initial part is actually called a renal arcuate tubule. And then the next part's called the straight collecting tubule. And the very bottom of the collecting tubule, where it dumps actually out into a minor renal calyx, remember that funnel-like collecting structure way down here at the bottom, that part is called the papillary duct. So there's actually 3 parts there, and I'm going to go ahead and put in parentheses. And you need not learn the names of those, but when you hear renal arcuate tubule or straight collecting tubule or papillary duct, just understand that they're talking about subparts of this collecting duct. So when we say the 4 main parts of a renal tubule, it's the 1, 2, 3, and 4 that we listed here. Now, in a nutshell, let me show you what's going on just as we look at this picture. Blood is flowing into this glomerulus and then out of the glomerulus. As that blood passes through the glomerular vessels right here, some of the fluid portion of the blood actually filters through the wall of these arterioles and through this internal layer of the glomerular capsule out into the lumen of the glomerular capsule, out where I've put these little red X's. That fluid is called filtrate. So as soon as we get fluid in here, we call that fluid filtrate. Then the filtrate flows down through the various parts of the renal tubule. And as it flows through the various parts of the renal tubule, the composition of the filtrate is modified. It's modified by some things being secreted into it. We have some secretion going on In other words, things are actively, physically passed into the filtrate. The composition is also modified by things being reabsorbed from the filtrate. A lot of different things are reabsorbed. Glucose is one of them. Glucose gets filtered into the filtrate, but most of that glucose will be reabsorbed as the filtrate passes through the various parts of the renal tubule. The biggest single thing, of course, that's reabsorbed from the filtrate is water. A lot of water is reabsorbed. So when we think about what's going on in one of these nephrons, and once you understand what's going on in a nephron, you understand the general physiology of the kidney. Think filtration, secretion, and selective reabsorption. Physiologists often say that urine is formed by filtration, secretion, and selective reabsorption. Those are the processes. Now, obviously, the filtration occurs up in the glomerulus and glomerular capsule part of this nephron. But the secretion and selective reabsorption processes occur in the various parts of the renal tubule. Does that make sense to you? Let's take a look at a model. Let me pull up a model that shows these things that we just talked about. And we'll put it right here. We just need to concentrate on this central section if we go to the overhead camera. Now, what we've got over here is a coronal or frontal plane section through a kidney. And if we take one chunk of that out, just make this piece right here out of the kidney and blow it up, we go over here to this next section. So basically we've got more or less represented here one renal pyramid, although it would have thousands and thousands and thousands of nephrons in it. Right here's a glomerulus. I don't know if I can get a small enough pointer here. Let me get a small pen. Here's a glomerulus, that little tuft of vessels, and then the glomerular capsule surrounds it. Proximal convoluted tubule, nephron loop coming down, going back up, distal convoluted tubule, and then finally the collecting duct. Now, one of the things I want you to notice here is that, see all these broken off stubs here, here, and then up here you can see it's not broken off. A single collecting duct will actually service several different nephrons. So every nephron has its own glomerulus and glomerular capsule and its own initial three parts of the renal tubular system, but several adjacent nephrons can share the same collecting duct. So there would be another distal convoluted tubule from another nephron connecting in here and here and here and here, so forth. Does that make sense to you? So this kind of shows you what's going on. Basically, right there where I've placed the pin is the corticomedullary junction. In other words, here's the cortex, and the majority of the glomeruli and glomerular capsules are out in the cortex. There are some that are down in the medullary region, but most of them are up in the cortex. And the medullary portion of the kidney consists mostly of nephron loops and collecting ducts coming down. And as that fluid called filtrate goes through this system, as soon as it gets down here to the bottom of the collecting duct, it drips out, if you will, of this collecting duct into the minor renal calyx, and at that point we call it urine. So its composition is changing as it goes down through the various parts of the nephron, but we still call it filtrate until it leaves the collecting duct. And once it dumps into the minor renal calyx down here, at that point that fluid is urine. Any questions about that general idea? Let's follow blood flow through the kidney. Let's go to a graphic that kind of shows the names of the vessels, and then we're actually going to look at a model and perhaps a drawing to see if we can clarify them. Starting here, coming off the aorta, we've got the renal artery that serves either the left or the right kidney. And it branches into a front branch and a back branch, if you will. And some of those, there's actually some branches that come off of those called segmental arteries. We'll not worry about those. But we actually have branches that go up through the medullary tissue that are called interlobar arteries. Each pyramid is called a lobe sometimes in the kidney. So interlobar would be what? Between the lobes, between the renal pyramids. And then those are actually interconnected to form arcuate arteries. Before we go on to the rest of this particular graphic, I want to leave the graphic and go back to the model for just a minute. And we'll just put the model right here. And stay on the overhead camera, and we'll take a look. Here comes the renal artery, and here you can see it branching. Here's an interlobar artery, here's an interlobar artery, here's an interlobar artery. So these are all interlobar arteries coming up between each one of these pyramids. The tips of the interlobar arteries are joined by these arches, they're called arcuate arteries. Okay? Coming off those are smaller vessels that go on up through the cortex. These smaller vessels are called interlobular arteries. So each one of these little vessels is an interlobular artery. And the afferent arterioles, the ones that are carrying blood into the glomerulus, are actually branches of those interlobular arteries. Now, if I go over to this segment here, which shows a much more magnified or high-power view, here's the arcuate artery. Here's the interlobular artery, and then each one of these branches coming off to an individual glomerulus is an afferent arteriole. Afferent arterioles carry blood into the glomerulus. Now, after going through the glomerulus itself, the blood comes out the efferent arteriole, as you can see on this one or on this one, and then that blood goes down through a little capillary network down into the medullary tissue. That capillary network is called peritubular capillary. Peritubular capillary, P-E-R-I, meaning around the tubules. So this forms a capillary network down here around the various parts of the renal tubule. After flowing through those capillaries, that blood returns into interlobular veins, like this one right here, interlobular vein, which dumps into an arcuate vein, and then that comes down as an interlobar vein. Let's look at the words again now and go back to that same graphic that we had up before on renal blood flow. So we were at interlobular arteries, and we've got afferent arteriole, then it goes through the glomerulus, then efferent arteriole carrying blood out of the glomerulus. And that blood then flows down through the peritubular capillaries. These are the small capillaries down around the renal tubules. They empty into interlobular veins, and then we just follow the sequence back out, arcuate veins, interlobar veins, and finally back into the renal vein, which dumps, of course, into the inferior vena cava. So it's kind of a complex little flow into particular named vessels. Now, I want to concentrate for a little bit on the glomerulus because I want to spend a little bit of time explaining how the glomerulus and glomerular capsule actually do their filtration thing. So let's go back to the overhead camera, and we said that the glomerulus was just a tuft of vessels. We had an input vessel, afferent arteriole, and an output or draining vessel, efferent arteriole. What I want to do is just grab a little segment of one of those glomerular vessels and blow it up down here. So we'll say this is that particular part of the glomerular vascular structure. Now, the vessels here actually have little teeny tiny holes in them. They're said to be fenestrated. So I'm just going to draw some little tiny circles, just like some sewer pipes that, you know, actually have holes in them on purpose to form part of the drainage system. However, these particular holes, although they allow fluid materials to pass through are not the size limiting factor in what can leave the lumen and pass through to become filtrate. What actually forms the size limiting factor are some vessels that are on, or some cells rather, that are on the surface of these vessels, and these cells are called podocytes. Now let me draw down here what a podocyte looks like. If you just kind of think of your hand with fingers, or think of an octopus, there's the nucleus. They have these little finger-like projections, and these finger-like projections interlock with the finger-like projections of adjacent podocytes, so that we end up with this kind of an arrangement, if you will, like my fingers are doing. But it's not a real tight arrangement. There's actually a little tiny bit of space in between them. These little finger-like projections are called pedicels. The name of the cell, again, is podocyte. Let me write that down. I always think of an octopus whenever I think of these, because it's kind of the way they, their shape reminds me of that particular animal. And so that's the name of the cell. The actual finger-like projection is a pedicel. So the name of the cell is podocyte, and the finger-like projection is a pedicel. Now, the space, the little tiny bit of space in between the pedicels of adjacent podocytes, these spaces are called filtration slits, and they are actually the size limiting factor. Now remember, these podocytes are actually on the surface of these vessels. So let me just draw little finger-like projections here, and we'll say those are the podocytes themselves with their little pedicels, and this is a little space right in here. I'm going to kind of color that red. So that's the space. It's the width of those filtration slits between the pedicels of adjacent podocytes, are you following me here, that actually determine the size of the structures that can pass from the lumen of this blood vessel out into the glomerular capsule itself. Now, blood cells are too big to pass through those filtration slits, so we normally do not get any blood cells infiltrate in a normal, healthy, functioning kidney. There's no blood in the filtrate at all. And the filtration slits are too small to allow most proteins through. Sometimes there's a trace of albumin that can get through. It's a relatively small blood protein, in fact. So cells and large biological macromolecules are not filtered. They will not pass through the glomerular wall to become part of the filtrate. What does pass through is mainly water, electrolytes, and small organic molecules. Did you have a question, Seth?
Yeah, I did. Can the podocytes change their shape and alter the filtration slit gap to change?
That's an interesting question, Seth. I've never heard or read about them being able to do that, but in certain disease processes, they can be altered. But I mean, I think your question is, can podocytes do this from a physiological standpoint too? I don't believe that's the case. When you get inflammation of a kidney, you get a nephritis going on, sometimes the inflammation will cause the filtration slits to get wider. And if you have that kind of process going on, obviously some things will pass through into the filtrate that normally would not. So in severe nephritis, you may actually have some blood cells passing through. And you may have larger proteins, globulins may be passing through with impunity if you have a severe nephritis. Now, I'm here to tell you that when you have blood in your urine, in most cases, it's not from the kidney. It's probably from a cystitis. You get the urinary bladder, when it gets angry, the lining of the urinary bladder bleeds quite easily. But it is possible to have blood in urine that's from a situation that we're just talking about, where we have a filtration slits severely widened by a nephritis-type disease complex going on. So at any rate, I hope I've explained the process here. What actually forms the size limiting factor is these little spaces in between adjacent podocytes, the filtration slits. Now when you're take a look in the Nomen Anatomica, the official name for those filtration slits is cytotrabecula, but everybody calls them filtration slits. Understand why. A little bit easier to understand, I think. Anyway, that's how the kidney goes about filtering the materials that it filters. Now, after filtration occurs, as the filtrate is passing down through the various parts of the renal tubule, remember we had a proximal convoluted tubule, then a nephron loop, then a distal convoluted tubule, and finally a collecting duct. As that filtrate passes down through there, some things are added to it, physically secreted by active transport into the filtrate. Other things are selectively reabsorbed, especially water. And I want to see if I can explain to you how the kidney goes about reabsorbing the water that's in the filtrate, because it absorbs a very large percentage. About roughly 99% of the filtrate volume gets reabsorbed during the trip down through the various parts of the renal tubule. So for every 100 mils of filtrate that we produce in the two kidneys, there's only 1 mil of definitive urine that results. And how can that be? Let's go back to the overhead camera for a bit. And let's take a look at, we'll just say this is part of the renal tubule. This could be a nephron loop down at the bottom end there, and a big long hairpin turn. The cells that actually, if I do a transverse section through the tubule itself right here and say, okay, this is the tubule, this is the lumen of the tubule, and these cells are simple cuboidal epithelial cells. One cell thick, kind of marshmallowy, globular, if you will. And I'm going to put an F right in the middle there. I didn't leave myself much room, but that F stands for filtrate. So the filtrate is right there in the middle, right in the lumen of that tubule. Now these cells have active transport mechanisms that actively transport sodium basically out into the interstitial fluid surrounding the tubule. So I'm just going to put some arrows. I'm going to write Na plus for sodium, and I'm going to write At in parentheses to remind myself that that sodium is being actively transported out. Now, if you actively transport sodium out into the interstitial tissue, sodium that's in here will do what? Sodium will move where? From the filtrate into the cell. And it'll just do that by what? Simple diffusion. Because we've got active transport mechanism moving the sodium out the other side of the cell, so sodium that's in the filtrate will move in this way, yes? Now, if you're actively transporting this sodium out in the interstitial fluid surrounding the tubule all the time, we're going to get a whole lot of sodium out here, yes? And what's that going to do to the osmotic pressure out here? It's going to raise the osmotic pressure. And so if you have a lot of sodium out here, then what's going to eventually follow that sodium? The water. So basically we actively transport the sodium out, and the water follows. That's how the fluid that's in the filtrate actually gets dehydrated, if you will. The water gets removed. Now, of course, removing the water from the filtrate does what? It moves that water out into the interstitial fluid, yes? Now, do you remember those paratubular capillaries that we talked about? After the blood comes out of a glomerulus, it goes into a paratubular capillary and goes down around the various parts of the tubule. That's the vessel that picks up the sodium and the water that's now out in the excess sodium and water that's now out in the interstitial fluid. So that's the general gist of how the kidney is involved, the renal tubule is involved in the reabsorption of water. Again, to boil it down to the simple concept here, sodium is being actively transported out of the filtrate, and the water passively follows by osmosis. And so that's how that occurs. Any questions there? Yes, Amber.
So, what happens when you pass kidney stones? Like, what causes the kidney stones to form?
Some individuals, due to their individual chemistry in the kidney, have a tendency for precipitates, materials to precipitate out of the filtrate and/or the urine after it finally dumps out of the collecting duct, and we call it urine at that point. Most of the time, most all of us form those, but they remain in most cases so small that they pass right on through the ureters and urinary bladder and urethra. In some individuals, though, they form such a large number of certain kinds that they tend to stick together and form these concretions that will form various renal or ureteral, cystic, or urethral calculi. And when those get lodged someplace, or when they're physically moving, if there's any size to them at all, then they can get your attention to the place where you're going to seek some medical attention in regard to it. I think I mentioned previously in our previous session that a ureteral calculus that's on the move, that's moving down through a ureter, sometimes is extremely painful to the where it'll put you down on the floor, kind of doubled up in the fetal position. It's that bad. It's quite often these things, depending on what they're formed of, will have sharp edges, and the mucosa of the ureters is pretty sensitive. So, Bryce.
And you also said earlier that sometimes the lining in the
Urinary bladder?
Yeah, and the urinary bladder will get inflamed and start bleeding. What causes that?
Most of the time it's a bacterial infection. That's the biggest cause of cystitis, which is the clinical name for urinary bladder infection, urinary cystitis. It can happen either in an ascending manner where bacteria are actually moving up the urethra, or it can happen from the bloodstream when we have a systemic septicemia, and bacteria in the bloodstream can actually get into the urinary bladder lumen and cause a problem. Most of them are ascending, and I think I may have mentioned before, if not, we'll mention it again when we talk about the reproductive system in future sessions, but females have a lot more problem with cystitis because the urethra is relatively short. It's just not very far from the outside world into the lumen of the urinary bladder, whereas males have less problem with cystitis as a group, simply because the urethra has to pass through the length of the penis, and as a consequence, males have a longer urethra. So if it's an ascending infection, which most of them are, it's harder for bacteria to get established up in the urinary bladder. Starley?
Going through this process, if you're drinking more water, you're making more urine only because the sodium level stays the same.
Yes.
Is that accurate? So when you're
Well,
taking in...
The kidney can, and there's a hormone that can actually control, it's a hormone produced by the adrenal cortex that actually controls the amount of sodium that's saved from the filtrate. So there's a couple of mechanisms. Matter of fact, let's talk about those mechanisms. In general, all things being equal, the more you drink, the more water you drink, the more urine you're going to form. And if you haven't been drinking much water lately, you're going to form a much smaller quantity of urine that has a higher concentration of waste products in it, right? Right. Before we go there, let's go ahead and take a look at this next graphic, because I want to make sure we get through this volumetrics thing a little bit. This is a little chart, and I've rounded off these numbers over here just a little bit to make them nice and even, but these are real close to the actual figures. Now, resting cardiac output is about 5,000 mils a minute, okay? And that's about 5 liters a minute. That means your aorta is pumping about 5 liters of blood a minute. 20% of that cardiac output actually goes to the 2 kidneys. So a significant portion of the blood going out the aorta is headed for the kidneys, about a fifth. That's about a liter a minute, 1,000 mils a minute. As that goes through all the glomeruli in both kidneys, about 100 milliliters, 100 cc's of filtrate is formed. So about 1/10 of the renal blood flow, 10% of 1,000 is 100. So about 10% of that renal blood flow becomes filtrate. Now remember, that's not blood, it's filtrate. It's water, electrolytes, and small organic molecules. No blood cells normally, no large proteins or other large organic molecules in it. As the filtrate goes down through the various parts of the renal tubule, about 99% of its volume is reabsorbed. So if you have 100 mils of filtrate total in both kidneys, and 99% of that is reabsorbed, then the definitive amount of urine that you're going to form. Now this is average size person, average state of hydration, about a cc or a milliliter a minute. So you do the math, that's 60 mils an hour, right? And then if you're twice as big as a normal person, a normal person is considered to be 70 kilograms, 150 pounds roughly, okay? If you're twice as big, everything on that chart is going to be doubled. If you're a 75-pound, youngster, everything on that chart is going to be halved, and so forth. Now, Starley, you asked about sodium. Obviously, if you're drinking regular water, it's relatively low sodium content. But sodium levels in the bloodstreams are somewhat controlled by a hormone called aldosterone. Let me just write the name of it on the overhead camera so everybody can see that. And under higher levels of aldosterone secretion, sodium is retained. So I'm just going to say retains sodium. It causes some changes in what's going on in the renal tubules so that sodium is retained. Okay? And when less aldosterone is secreted by the adrenal cortex, less sodium is retained. And in general, the water does follow the sodium. So there's a general, if you retain the sodium, you tend to retain water as well. So anyway, that's one way that the kidney actually controls body water content. If we go down to the distal part, let's stay on this overhead camera for just a minute. If this is the collecting tubule, there's a hormone that influences what's going on in the collecting tubule called ADH. Now, I don't know if you remember from your reading on the endocrine system, but this is an acronym that stands for anti-diuretic hormone. ADH, anti-diuretic hormone. Now, diuresis, this word diuresis, implies urine formation, okay? So a hormone that's anti-diuretic would be a hormone that helps lower the amount of urine formed. ADH is released, let me just go right up here, running out of room. This is released from the posterior pituitary gland. I'm going to write P-O-S-T-P-I-T. Released from it's actually manufactured, you'll recall, by the hypothalamus, but it's stored in and released from the posterior pituitary gland. It's released when we are dehydrated. It's a hot day, you haven't drunk any water. You can bet that the anti-diuretic hormone in your bloodstream is going to be pretty high. What that does, this hormone actually goes down here and influences the water permeability through the walls of the distal or the collecting duct. Now, the collecting duct wall is usually relatively impermeable to water passage. So if we've got any water that's still in the filtrate at this point, cannot get through the wall under normal circumstances. Remember, we got a high sodium content out here in the interstitial fluid, so there's osmotic pressure. It's trying to draw that water right on through that collecting duct wall. But the collecting duct under normal circumstances is impermeable. Under the influence of ADH, however, the wall of the collecting duct becomes relatively permeable. And when it becomes permeable, because of this high osmotic pressure, lots of sodium's out here, that water will go out into the interstitial fluid. Now, if you've got a lot of water leaving the last part of the filtrate, what's that going to do to the total amount of fluid going down here to form urine? It's going to reduce it, right? So we get less urine formed as a result, which actually translates, as far as the body's concerned, making fewer cc's of urine means more water is being conserved by the body. So if you're in a dehydrated state, your body knows that it needs to make a very concentrated urine and a relatively small amount of urine, so that a lot of waste products are in that, and it basically does that by releasing ADH, which goes down here and affects the permeability of that collecting duct, and a lot more water is pulled out of the filtrate right at the last part than would normally be under other circumstances. Does that make sense? All right, a couple of other things we want to talk about. Let's go to a graphic here, and I mentioned this a while ago, but I don't know if you caught it. I doubt if I made the point very well. Glucose is normally filtered. In other words, glucose in the bloodstream will enter the filtrate. It'll become one of the molecules in the filtrate, one of the small organic molecules. But normally, we have no glucose in definitive urine. Now, why is that? Susan?
It's because the body goes to a certain level of glucose, and then it will filter out the small filtrate into...
Well, okay, we're saying we got glucose in the filtrate.
Right, but then it goes out with the water because it's a small molecule.
Actually, glucose is actively reabsorbed.
Okay.
So it's not following by an osmotic process or a simple diffusion. It is actually being selectively reabsorbed. It's actively transported out of the filtrate. So let's say I go down to the grocery store this afternoon and buy a pound of candy. And of course, it would take what? At least a quart of ice cold milk along with that, right? Do you suppose that and if we did a urinalysis on me after I did all that in about an hour, do you suppose I'd have any glucose in my urine? Probably. Because if you have a lot of glucose in the filtrate, you can exceed the ability of the active transport system to reabsorb it. So, if I'm an uncontrolled sugar diabetic and I have extremely high blood levels of glucose, I'm going to get a lot of glucose in my filtrate, and some of that glucose is going to spill over and not be reabsorbed. And so it'll get into the filtrate, and not all of it will be reabsorbed because there's so much of it. Does that make sense to you? Mm-hmm. I mean, if it's your job picking out the bad tomatoes on the assembly line here, and there's 5,000 bad tomatoes coming down for every good one, you're going to have some bad ones slip by. Whereas if most of them are good and there's just an occasional bad one, you can get it. It's a poor illustration perhaps, but that's the idea. You can actually exceed the ability of the reabsorption mechanisms to do their job. Seth?
Why is the glucose filtered out if it's just going to be reabsorbed again?
It gets filtered out just because it's a small molecule, and it just kind of goes with the flow. Okay? Remember, water, electrolytes, and small organic molecules just get passively filtered. So that's the reason. There's no purpose for it being filtered other than it just happens to be a small organic molecule. Let's go to a graphic here. I want to define renal plasma threshold. It's the minimum concentration of a substance that results in excretion of that substance in the urine. When I say minimal concentration, we're talking about how much of that stuff has to be in the plasma itself in order for it to start showing up in the urine. And for glucose, which is what we've been talking about, if you've got 180 to 200 milligrams of glucose per 100 mils of plasma, then it'll start showing up a little bit in the urine. If you get your blood levels of glucose up this high, then all of that glucose cannot be reabsorbed from the filtrate. A diuretic, we mentioned antidiuretic hormone and used the word diuretic a while ago. A diuretic is a substance that increases the volume of urine formed. Now, the most obvious diuretic is water itself. You drink a lot of water, you have to urinate a lot because you're forming a lot of urine. You don't drink very much water or other fluids, the total amount of urine that you will form will be much less. But there are a lot of other substances that serve as diuretics as well. Caffeine has some diuretic properties. Lasix is a common diuretic that's sometimes given to individuals, that have a big fluid buildup, a big edematous type situation that really will tremendously increase the total volume of urine formed. Let's take a look at this graphic then. Any substance that increases the volume of the urine formed can be considered to be a diuretic. They do lower the blood pressure because if you lower the total volume of blood by forming more urine, the blood pressure is going to go down. Does that make sense? Susan?
Is that the basis of some of the medications that they give for blood pressure? It's just a diuretic to
That
control?
is, in fact. Well, some of them are. Some of them are diuretics. Others, there's some other mechanisms by which certain blood pressure medications work. Starley?
How do diuretics work? Like, are they making the sodium not stay on the outside, or?
Well, how can I have more cash flow? I could get more cash flow because they give me a raise. I could get more cash flow because I finally get all the kids moved out of the house, and so they're not the financial drain. You know what I mean? I can get more cash flow because I quit spending it foolishly and start using it a little more wisely and so forth. There's a lot of different mechanisms. There's lots of different diuretics, and some of them actually act on the various parts of the renal tubule in different ways. Some of them act on the amount of sodium that's retained and so forth. Remember, water follows sodium. So if we're retaining sodium, we'll retain water. If we're losing sodium, we'll lose water in the provisional urine that's being formed. A couple of other terms I wanted to define real quickly, if we can go to the overhead. Uremia. This is a buildup. Anybody know? This means something in the bloodstream. What is it?
Is it uric acid?
Not uric acid.
Urine.
Remember this compound? End product of protein metabolism. What was it called?
Urea?
Urea. Uremia is simply a high concentration of urea in the bloodstream. We've run out of time. Next time we'll be talking about the male.